Okay, yeah, it's just 2 after 7 now, so I think we're going to get started with today's talk. If everyone is everyone been welcomed from the waiting room? Great, perfect. So this will be the first talk of this semester, our Science Under the Stars series, and we have a real treat. Starting us off, Mackenzie Johnson is an esteemed colleague and a super genius here, working on working with computers to answer some bigger questions in evolutionary biology. And so it's my privilege to introduce her. I'm Tristan Kubik. I'm stepping in for Kyle this semester as host. I'm very privileged to be the host of Science Under the Stars this semester. And so enough about that. Without further ado, I'll welcome Mackenzie and let her get things started. Go ahead, Mackenzie, take it away.
Okay, great. Thanks, Tristan, for your introduction. And thank you all for being here today. So like Tristan mentioned, I'm a computational biologist. And what that means is that I use computers to try and understand biology a little bit better. So today, what I'm going to tell you about is how we use computers to teach us more about the natural world around us. Okay, so I'm assuming that all of you that are here have some idea about what a biologist is, but I'd like you to take a second and think, what is a biologist and what do they look like when they're doing research? And a lot of you, the image that probably comes to your head is someone out in the field doing something with biology. And so that's the case for many people. It's really common to go out into the field and observe the natural populations all around us. So here's an example of a project when I got to go out into the field and count the different species of plants that were found within this native tallgrass prairie in Kansas. And so that was like an important survey to find out what was living in that area. We also have grad students in our department that are currently do a lot of field work. For example, Allison Davis is a grad student, and she studies fish and some of their behavior. And here she is surveying a site through our Ecolabs program somewhere outside of Austin, looking to see if mosquito fish are present or not. And we also have biologists that study natural responses to different disturbances. So Whitney Bear is a grad student who studies how different flowering plants respond after wildfires. So we also have people that go into the lab to, go into the field to collect samples from natural populations. So for example, Annalisa Shields Estrada is a grad student in our department who studies tree frogs across Texas. And so she goes and she samples them from all different areas, brings them back to the lab, and then performs experiments on them to see how their coloration impacts their ability to withstand different temperatures. We also have Thomas Marshall, who studies rat snakes in central parts of America. And so he collects snakes that he finds all through Kansas and Texas and brings them back to the lab to collect their DNA and figure out how they're all related to each other. And I should mention that Tom gave a talk earlier this year for Science Under the Stars, and it's available on YouTube if you would like to find out more about the snakes of Central Texas. So these are all people that go out into the field and do field work, but we also have people that do work in the lab. And so this might be processing this data that they bring in, or it could be doing experiments to generate new data sets. So this is an example of an experiment that I participated in, where we were looking at how different fungi were able to remove fuel from soil. So that way we could learn more about maybe some natural bioremediation experiments. And we also have a grad student here, Julia York, who studies electric fish and how they survive in such extreme environments. And she will look under the microscope and study how different molecules make this possible. And she'll be giving a SETS talk later this year. So make sure you come back out for that. Yeah, so you might envision that most biologists are working either in these two environments. But for me, all of my work is on the computer. So computational biologists like me either analyze these existing data sets or we simulate our own with computers. So this is my everyday life as a computational biologist. Here I wrote some simulations to generate some data to ask a question about biology. But we also might use databases like this database, the NCBI database. So one great new thing that we do in science is the standard of like open science. So now it's best practice for researchers to go ahead and upload their data and make their code available so that way other people can either re-examine that data or use it in a different way. So I do both of those things. And occasionally I get to go out and do my version of field work, which is me just traveling somewhere to sit in a room with other computational scientists and I'll stare at our computers together. And this is actually a very exciting time to be a computational biologist, and that's because we have a lot of data. So this database that I was referring to here, this NCBI GenBig database, has seen a big increase in data. So in this plot, we have time from like the 1980s to present. And then on this y-axis, we have the number of sequences that are found in that database. And as you can see, within the past few decades, we have like greatly exceeded the amount of data that we've ever had before and are quickly approaching 1 billion sequences. So it's not just the data that we have readily available. We also have seen like this rapid growth in the computational resources that we can use to like do different science. And so again, on this x-axis, I have time in years from 1993 to present. And then on the y-axis, I have a measure of supercomputer power, so in flops. These flops are just the number of operations that a computer can do in a second. And when you look at the growth that we've seen in all the supercomputers in the world, you can again see that our compute power has grown like exponentially, and we really have no shortage of computational resources now to do work. So I'm, as a computational biologist, really interested in using those resources to build tools that will allow us to better explore natural populations. And so now I want you to just think, what is a population? Well, let's think about a specific area. So for example, we can think about Texas, where most of us probably are. And in Texas, you might have a bunch of beetles. And so I'm just going to use these little cartoon beetles, in this case, that are either red or orange. And so we would consider maybe these a population, which is just a group of individuals of the same species that are all living together in the same area. And so that was an example of a population, but we have real populations that we see in nature. And when we look at these natural populations, we can observe a lot of variation between different individuals. So when you take a look at this population here, these are all beetles of the same species, but you can see that there are a lot of differences between those individuals. And so one of the first things you might notice is that there's variation in their color. So you have some individuals that are very dark, while some are duller. You have variation in body size. So you have some small beetles and some quite large beetles. You also have variation in the number of spots that these different beetles might have, and in the size of those spots. And there's probably many more. We could probably find a lot more variation if we looked really closely at these beetles. And so this variation that we see in populations is really important to the survival of that population. And I'm going to tell you a bit about why that is. So again, I want you to think about this population that we have with these red and orange beetles. And this variation that we observe between individuals, we often refer to as diversity. And so this diversity, or the amount of variation that you have in a population, will vary from one population to another. So we still have our population 1 with these red and orange beetles, but we might also imagine a population that has green and blue beetles. And so these populations have the same number of individuals, but population 2 is more diverse than population 1, right? There's a lot more differences here than you have in population 1. So these populations don't just exist in a vacuum, right? They exist in real life. So you can imagine that they live on something, and they might have a natural environment that has a red background that they're surviving on. And when you look at this, what I really want you to see here is that with population 1, these red and orange beetles are really well camouflaged to their natural background or their natural environment. Whereas population 2 has these green and blue variants that stand out a lot against that natural background. And the reason why you would care about being well camouflaged is the fact that these beetles have predators, right? So there might be birds that want to eat the beetles. And when they go to both of these populations, they'll be able to see more or less of them, right? So with population 1, they might be able to find a few of them if they look closely, but with population 2, they will be able to see a lot more beetles and be able to eat a lot more of them. So in this case, we would say that population 1 is well adapted to its natural environment. So over many, many generations, this population has been living on this red background, and the red and the orange individuals have done the best, they've survived the longest, and they've had offspring. So now this population is adapted to this environment, which is great, but we know that environments are subject to change. So if you think about long-term, we have big changes in the climate, but we also have like seasonal changes. So you can imagine that maybe these populations, even though they're naturally found in some place with a red environment, the environment might switch to more of a green background. And in this case, you have suddenly a shift in advantage to this population 2 that has these green variants that better blend into their background, while population 1 has no variants that would match that background. So they're more likely to get eaten here. And you can imagine that the environment might change again. And again, in this scenario, population 2 would have the advantage. So what we're seeing here really is that population 2 is better able to handle more environments. And so that really, what that really shows us is that when we compare population 1, which is less diverse, to population 2, which is more diverse, we would say that population 2 is more resilient. And that is, what that means is that it's able to survive in a lot more backgrounds, right? The population will thrive long-term. And so it's for this reason that we really care about diversity in populations, and we would like to maintain it. And so we are often worried about populations with low diversity because they are at risk of extinction. And so populations that have low diversity might have few individuals, like this example here where they only have 4 individuals, so they could very quickly all get taken out by some event. Or populations that have a lot of individuals but little variation among them. So they're essentially all the same and they're all susceptible to the same things. And when I'm talking about these populations that have low numbers or little diversity, a thought that might come into your head is endangered species. And it's for these reasons that we are concerned about these species. We're worried about their long-term health and their ability to remain in our ecosystem. And you might think, oh, it's okay if we lose a few species every now and then, but all the species that we live with play an important role. We're all interconnected. And so it's really important that we maintain these different populations and the natural diversity that we find all around us. Okay, so when we think about this actual population and this little cartoon population that I'm showing you, you might see that most of the diversity that we're seeing here is visible, right? So in this case, you have diversity that's visible in color and patterns and their spots, while in this population, it's just color. But we can see all of these, all this variation between individuals. But the reality is most diversity that exists in natural populations is not visible to us. So when you think about one of these individual beetles, those beetles are actually made up of a bunch of cells which contain DNA. So DNA is the microscopic material that has all the instructions that make a beetle or make you. And when we look closer at this DNA, what it really is is these really long sentences of 4 different letters. And this, doesn't mean much to us, but it's very important. And in computational biology, we have tools that allow us to find the genetic differences between individuals. So now when I'm talking about genetic diversity, I mean not differences between individuals in their appearance, but really in their DNA. And so for the same reasons that the visible diversity that we see is important to the survival of a population, this genetic diversity is also important, and we want to maintain it. So one really cool thing is that your DNA holds a lot of information that we can extract using various tools to get information about how that population has lived in the past and how it might be impacted in the future. So for example, when you look at the DNA of DNA of individuals in a population, you can use various methods to figure out what was that population size in the past, like before we were even around, and what would that population size be in the future under different scenarios. So there's a lot of information in DNA, and what computational biologists do is try and be this tool that allows us to extract the information from the DNA that exists in the world. And so, I've told you that we're concerned about populations that have little diversity. So you might be wondering, how do we uncover kind of the health and the history of a population that we're interested in? So for this, I'm just, again, thinking of a population here as just kind of a mix of dots, but you can imagine them to be whatever your favorite species is. So these are all just individuals that vary in some way. And so if we want to find out more about the population, we need to study the genetic variation in that population. And in reality, we can't go out and we can't take a whole entire population and bring it back to the lab and study it, because then there would be none left. So what we have to do is we have to just pull samples from that population. So by that, I just mean in this population, we select a few individuals and we put them together and we take them back to the lab, and that's what we study. So we hope that this sample is representative of the natural population that we're pulling them from. And hopefully you'll do this randomly, but you might also have a method for the way that you're sampling the population. And once you have these samples, you can take them with you, and we can use various technologies that are available to us to extract information from them. So for example, we might use sequencers, which are big machines, which take those tissues and from the DNA in those tissues, give us the actual sequence of these individuals. So now for each individual that we see, we have its DNA. And what this DNA is again, for these individuals, it's just a long string of these letters, these 4 letters. And we like to align them in this fashion. So that way you have these 4 different samples, and all of their DNA is lined up. So that way the same site across individuals is in the same place, right? So we want to look at just a part of the DNA, and we want to compare the same part of the DNA between all of these these individuals in our sample. So we have them lined up like this, and we have these really long sequences, and now we want to compare at each site in the DNA how much variation is there. And so it's probably hard for you to see from here, but you'll notice if you look closer that there is variation at some sites. So for many sites, it's just the same letters found in each individual in that sample. But at some sites, we have variation, and there will be, perhaps it'll be 2 different letters there. So for example, for site 2, we have one individual in this population has a C while the others have G, and so on and so forth. And so this variation is really where we're getting our information about the population. And just to simplify this, so instead of having to look at all of these letters, let's think about just the cases. So let's turn this into like a simple black and white example. So for sites that have a conserved letter where they all are matching, we just visualize that as black. And for sites where there's variation, we might have our one sample as white and the others that are matching as black. So this is just for us easier to look at and to see where the variation is. And again, I just want to like emphasize like how big these genome sequences are. So for humans, if you were to sequence the full genome, it's about 4.6 billion letters long. So that is absolutely massive. It's too much to fit on a computer. So if we want to actually study these populations, we need to reduce the amount of data here that we have here without reducing the amount of information. So what we can do is we can just simply remove those sites that are constant, right? Those sites that were black all the way down. And then we get a little alignment that looks like this, that is black and white, and it has all the information about how this population varies. So I'm going to show this kind of figure here a lot through my talk. This is the data that I work with. It is just simply like black and white images like this that are alignments that just show where in the DNA individuals in this sample vary. Oops. Okay, so in the field of biology, we have a long history of developing tools to extract information from these alignments. And so what these tools are they are built on like mathematical frameworks that are based on the things that we know about biology and how populations work. And then to apply these frameworks, we develop computer programs that use this math on this data. And what these tools allow you to do is extract information from this alignment. So now instead of this alignment, we might find out information like how these individuals are related to each other, information about how big this population was in the past, or we can also find out things about like how quickly new variants or like more diversity is being introduced into the population. So this is really great. Like there's like decades of work built into this, but these tools have limitations. So they work in cases where we know a lot of information. So for example, these traditional methods work well if you have like a model organism. So model organisms are things like mice or Drosophila or C. elegans, these things that we've, as a scientific community, spent a lot of money and resources on understanding really well. So that way we can learn things from those systems that we understand well and apply them to other systems. So we understand in these organisms really a lot about their biology and how they work. But if you don't have a model organism, then you might need to generate a lot of data if you would like to study a new population. And generating all that data will probably cost you a lot of money. So we've kind of come to this crossroads where we have all this data, we have all this power, but we don't have the methods that we need to be able to work. So these traditional methods are kind of currently our limitation. And when I like refer to these methods in the future, I'll just kind of continue using this little schematic of the magnifying glass on the DNA. So now you might be wondering, okay, there's limitations to these tools. How do we actually study populations when these methods won't work? And the answer that we've found in the past few years is machine learning. So I'm just going to quickly explain the kind of paradigm of machine learning and what we're doing here in relation to traditional tools. So with our traditional tools, we have data and we have rules. So what that means in the biological context is that we have those alignments, right? Those black and white images, and we have rules. So we have an understanding of the math about how these systems work. And so we package those rules and that data into programs. And out of those programs, we get an answer that we're looking for. Like for example, a population size, the number of individuals in that population that exist at some point. But machine learning fills this gap when we don't know those rules. So with machine learning, what you do is you have your data and you have labels for those data, right? So you know the answer for that alignment. You know how many individuals actually live in that population, and and the alignment that goes with it. And so you give those labeled alignments to the machine learning algorithm, and then the machine or the computer will actually learn the rules, right? So the computer is essentially learning how biology works without knowing anything about biology. So this is very exciting. And just to give you kind of an example of how what we're really doing here, I'm going to talk about this specific case. So I'd like you to imagine that you are some sort of software engineer working at a company that's interested in self-driving cars. Now, if you want to build a self-driving car that's like safe to go on the road, it needs to be able to figure out what objects are near it on the road, right? So when you look at a road, you might find a bunch of different things, right? You might find cars, you could see buses, and you might also see people riding around on bikes. And so it's really important that you're able to identify all these things. So machine learning has really helped in applications like this. And the way that it works is that you have a bunch of images of cars, trucks, vans, bikes, whatever else you might expect to find on the road. And you have those images labeled. So you have a picture of a car and you have a label that says car. And what the computer does is it first learns about the features of this object. So for a car, it might pick up on the fact that most cars have headlights, or most cars have wheels and doors, and so on and so forth. So it will learn the rules about what makes a picture a car, and then it's able to classify that as a car. And what's going on in the background is like some complicated stuff that it's happening. You might have to tell it a few things, but basically the computer is automating all this learning for you. So this is very exciting. And within the past like 3 years, biologists have really started to adopt these machine learning tools to study like natural populations, which is very exciting. So we can use these images that I showed you earlier, which are alignments that show you the natural diversity that exists in a population. And when we look at these images, they don't really mean anything to us, right? They, you know, we see that there's differences between them, but we don't detect any patterns. But you can imagine that computers are really good at picking up different patterns and using different math to find things that we'll never find. And so we can take these alignments, we can train a computer to learn about biology. And then out of that computer with our machine learning tool, we can get the information that we want from that population. So in this case, it might be the population size that sample came from. Okay, so to actually do machine learning, you need a few things. You need softwares, so like computer programs, and you need computers to run them on. So luckily, because biology is using tools that have been developed for a long time, there are a lot of really nice softwares that are available from like computer scientists and such that allow us to basically just apply their frameworks to our data. And we also use supercomputers. So in order to do work like this, like you need more power than you can get on the computer that you have in your own home. So we use these things that we call supercomputers. So this is an example of a supercomputer that it takes up a whole room, and it's just stacks and stacks of computers inside here. And so this gives us a lot of power to do what we need to do. And I don't know how many of you are aware, but UT Austin has really great computational resources. So at the Texas Advanced Computing Center on campus, or TAC as we call it, we have some of the fastest supercomputers in the world, and we have the fastest supercomputer at any university. So this is like a really great place to be doing this work. And we're like very excited about these resources available to us. So this is maybe the most exciting day of my graduate school experience. And I guess my version of fieldwork was the day I got to go visit TAC. So this is just one of the supercomputers. It's not the full thing. This is just the front of it. It takes up a huge room. So this is Stampede 2. This is what I do all of my work on. And inside of this are just computers. And if you look, so this is just there's a fancy case on the front to make it look nice. But if you look inside, it is just really stacks of computers on top of each other. So that way we can do a lot of really powerful work. Okay, and so in order for a computer to learn how to take this and give it this, we need to supply it with labeled data, right? So we need to tell it, show it a ton of these with known answers, so that way it can learn the rules. So that's exactly what I'm referring to here, where we have these alignments with the information about the diversity in the population, and then a label of the true answer in that population. And to do this, we need a lot of examples. So for my work, I normally use at least 60,000 simulations to train one model. I've told you earlier that for most species, or many species, we cannot use the traditional tools that would allow us to label this data, right? So we have this problem of we can't apply traditional tools to this data, but we also don't know the truth. So how can we train a model to, or how can we train machine learning models to do that? Well, the answer is we use evolutionary simulations to generate data like this, where we have an alignment with a known value of what it came from. And so what simulations are they're just imitations of a natural system. So in this case, we just pretend like we have a population, and we reduce the population down to a simplified model. So just to its essential elements, and then we generate data that we think that would come from just that, like the basic imitation of that system. And so I'm sure some of you have heard this quote, all models are wrong, but some are useful. And that's really just getting at the fact that models are never going to capture the full complexity of the natural world around us, but that we might be able to we might be able to build models that capture the essential parts of a system and are able to be useful for tasks like this. So what I'm doing as a computational biologist is working to make these evolutionary simulations more useful for machine learning applications. So these simulations are a model, and just by nature, models require that you have some simplifying assumptions, right? We don't know everything that happens in a natural system, and we also would never be able to fully capture that with a computer program. So we make some simplifying assumptions and try and get as close as we can to that natural population. And to actually train these models, we do need this realistic data, so that way we can apply them to our natural populations. So what I'm doing is evaluating in what cases are our simulation models useful for our machine learning task. So really what I'm asking is, what are assumptions that we made in our models can we use to generate realistic data? And what assumptions cause us to produce unrealistic data? So really with machine learning, what we're hoping to be able to do is ask old questions in new populations. So where our traditional tools fail, hopefully we can use machine learning to learn more about these systems. And by by applying these to new systems, we'll be able to broaden our understanding of the natural world, hopefully. So there's a lot of things that we've never been able to study before at the genetic level, and this will hopefully be one way that we can start to sample the full diversity that exists in the world. And so I hope today that I've kind of convinced you that computers can improve our ability to understand biology. So biology is really a collaborative science. And it's exciting to be at a place like UT in our integrative biology department, where I can work with these people that I was talking to you about earlier, that go out and collect data from natural populations or generated in the lab, and use help build tools so that way we can learn more about the data that they've collected. So with that, I'd like to thank the organizers and you all for attending my lab and the sources of funding for this work. And I'd be happy to take any questions.
Yeah, so now we thank you, Mac, for that outstanding talk. Very interesting stuff. And we can start taking questions in the chat. If you want to just unmute yourself and ask questions, I think you're also free to do that. Yeah, this is always my favorite part because everyone has such interesting questions. I think there was one in the chat here, Mac. Let me take a look here. Isabel asked, how often do you collect data? Her Alexa, her 9-year-old daughter, I think, is asking this.
Well, I generally don't go out into the field to collect data, but sometimes I go with my friends who seem to spend most of their summer collecting data. But I also will generate my own data on the computer, I'd say weekly. So yeah, kind of always collecting my computational data.
We have another question here from Percy in the chat. Percy said, thank you for the presentation. Can you give an example of a problem that computational biology has solved or a recent project you were involved in?
Yeah, so the work that I do is really trying to understand recombination rate variation in populations. So how quickly the diversity in that population is being mixed between individuals to create like novel variants. But maybe like a more exciting, like satisfying answer to that is there's this exciting competition that happens every year where a bunch of scientists try and figure out the structure of different proteins, which is a very hard problem. And like last year or two, like one of Google's groups submitted a very fancy machine learning thing called AlphaFold that you might have heard about in the news. And that was able to like outperform any biologically inspired model that we already had. So there's been a lot of recent applications to biology and they seem very promising.
We have kind of a deep question now, which was, how did you and those around you become computational biologists? And then there's a few follow-up questions, but I think I'll let you answer that one first, Mac, and then we can get to some of these meatier ones.
Yeah, so I became a computational biologist because I had a math class that I thought was really cool, where they were using it was differential equations, and they were giving an example of how you can use equations to like model how a population changes over time. And I thought that was so cool that I went and switched my major to biology that day. And then ever since then have really just kind of stuck to computational biology and teaching myself to program. And I have tried other types of biology, like in the field and in the lab, but I definitely prefer being on the computer.
So I think this second question then is kind of related to that. So you mentioned, you went and switched to biology that day. So what were you doing before that, before you took that course?
I was studying chemistry, but I didn't know why. So I was just taking a lot of math classes and finally found a way that I could use math that was enjoyable.
Oh, very cool. Following up on that one, so if you could start your career over, would you do it differently? If you did, how would it be better?
Oh, yeah, I don't know. I like the path that I have into biology because I think that it gave me a lot of tools that you don't get in kind of a standard like training for biology. And at UT, like I've been able to do a lot of really cool programs in the like statistics department to learn even more about those tools. So I don't think that I would do it any differently, although maybe it always feels like you could have started earlier. But I think there's plenty of time to learn more.
I have a less oh, here we go. Here we have another question here. This is another thanks for your awesome presentation. And then Miles, 11 years old, is asking, what do you mean about models? Miles wants to know exactly what a model is.
Yeah, so a model is basically just a simplified representation of something, right? So I guess maybe an example that you might have seen would be like a model car or something, where the real cars are quite big and they have a lot of machinery in them, but model cars are small and they just have the kind of the essential parts that make it look like a car. So yeah, with biology models, what I do, instead of looking at a population and seeing how, you know, all those individuals in those populations have relationships to each other and they have a life that they've lived and all these things, instead of looking at that, I really just look at just one part of their DNA and forget about all those things that those individuals do in their life. But I just look at how their DNA survives across generations. So very, very simplified view of the natural world.
I think that was an excellent answer, Mac. Thank you. So I had a quick question for you myself, actually, Mac. So when we're talking about models and these machine learning programs that are learning the rules of how a system works, how long does it take for a machine to figure those rules out? Do these programs last days, weeks, hours? How long generally? Or is there a lot more to it than that?
Yeah, I mean, the amount of time that it takes to write the code is maybe a little bit longer, but when I have all my data ready to go and the model set up, I can, you know, train a machine learning model to estimate recombination in, you know, an hour. So for my applications, it can be quite fast, but there are applications that might take weeks or so to learn. So like the example that I was talking about earlier of AlphaFold, I think it took, you know, a very, very long time to run. And the reason why, you know, folks like Google are able to do that is because they have unlimited resources and lots of money to do that.
Mm-hmm. There's another question in the chat here about what tools and workshops or clubs or resources are there on campus, I'm assuming UT's campus, that you would recommend for grad students that are interested in getting into computational biology?
Yeah, I think there are a lot of workshops, but those might be mostly at the grad level, but there are several classes, especially that are offered through like the Department of Statistics and Data Science that have to do with computational biology. And I think there's one in like that's listed in biochemistry, but so there are some courses, but also if you're like interested in research, I would just reach out to like a computational biology lab if you would like to learn more, because there are generally ways that you can get involved and you don't have to know anything about it. When I joined my computational biology lab in undergrad, I didn't know anything about biology. And I certainly didn't know about computers. So I just kind of showed up and they taught me from there. So yeah, I think there's a lot of computational biologists at UT. And I'm sure there's probably even like clubs at the undergraduate level for sure.
All righty. Last chance for any questions, any outstanding questions from anyone else here. We'll give you a little bit of time to type something in. Oh, great. Here's another one. This is another kind of existential question, I guess I would say, for you, Mackenzie. What makes a computational biologist? It's kind of a new, fresh field. So is it the courses you take that make you a computational biologist, a program you enroll in, just the aspects of your research, your programming? What do you think? I think what we're getting at is, what would you call a computational biologist, Mackenzie?
Yeah, I would think that most people that do biology are in some way a computational biologist. So even if you're not spending all day on the computer, I think even the people that work in labs or in the field would agree that they spend a lot of time working with their data on the computer. So I think, there are a lot of ways to be a computational biologist. I definitely identify strongly as a computational biologist because that's all I do is computational. So everything I do is on the computer. But yeah, I mean, the field has been around for a while. It really used to be more theoretical, but now we have a lot of data that we're trying to deal with. So I think that the field is definitely exploding, and there's a lot of incentive to incorporate computational biology into your work somehow. But yeah, I am just in the normal integrative biology program, but I just take a lot of extra classes in stats and computer science, and spend a lot of time learning new programming language.
Hey, we'll give folks a little more time to ask some questions too if they want. Again, thank you, Mac, for an outstanding talk and such eloquent answers to some of these really deep questions. Simply outstanding. It was a very rewarding talk. What a great way to start off the semester.
Yeah, thank you. Thank you all for coming out.
Yeah, if there's no more questions, I'll just give a quick update. Our next meeting will be the second Thursday of next month. And that talk will be delivered by another graduate student, of course, in our grad program, the outstanding Britt White. She'll be talking about lizards, legged and legless lizards. That's sure to be another humdinger, so definitely come out and visit us for that talk as well. It'll be a virtual talk as well, so we'll meet right back here in your own living room and deliver science straight to you from there. And so yeah, if there's no more questions, please join me in thanking Mackenzie again for a great talk here. Oh, we have one more question here in the chat, which is, have you helped others in the biology department with your projects?
Yeah, and I've often collaborated with people that maybe go out and do the hands-on biology. But yeah, and it's my goal that the programs that I develop, I could get data from the various people in our department and apply it to that. So yeah, hopefully there will be even more opportunities to collaborate there.
Absolutely. Great. Yeah, many thanks. All right. Thanks again, everyone, for coming out, and join us next month for our talk by Britt White.